Jim is one of these people who has got a long and illustrious career working in systems biology. And the reason I've invited him to give this talk is because as a group, as our Pharmacometrics Africa group, we've been doing more PBPK modeling over the past couple of years. We're quite strong on population pharmacokinetics, but these kind of bottom up or very systems biology approaches are quite new. And one question we get asked almost every time is, when do you go for QSP? When do you use PBPK? Is QSP really just PBPK with a few more parameters? What are the sort of pros and cons? And these are the kind of questions that frequently come up in our group. And Jim has worked in numerous companies, including establishing them and rising up to very senior level. And as he was saying in the backstage just now, has a few professional scars on his back. But I've asked him if he could really draw from his experience on these systems biology models, and then we can go into a time of question and answer. So Jim, over to you, and thank you very much.
Thank you, Katrina, and thanks for the opportunity to be with this group. It's very exciting to chat with you. Yeah, I've been working in quantitative systems pharmacology for about 25 years. My background is in chemical engineering, chemical and electrical engineering, I guess, always with a focus on control theory, systems dynamics, how things change over time, being able to model those. And the key thing is, as Katrina mentioned, Professor Waite said that she said, can you like talk about how you might choose PBPK or QSP? And you could go expand that to NCA, PK, POP. I think the people in this group know a lot more about POP PK than I do. But I do have some things to say about QSP and PBPK that I hope are useful. This is going to be somewhat idiosyncratic, and I'm going to share some observations. We will get to some models that we use. I'm going to start out with a non-small cell lung cancer QSP model, but we'll try to give you a basis for why we're doing what we're doing, how we do what we do, and some of the decisions involved. So now, the first company I worked with in this area, I was working for DuPont making Teflon, and I was hired by a California startup to work in modeling physiology. And the company's name was Intellos. It's no longer with us. And it was started by a group of people that had a foundation in decision analytics. The CEO and the chief modeling guy, both, the CEO was a PhD graduate of Stanford University's School of Engineering Management.
Hi, Jim. Hi,
Hello?
Jim. Sorry.
Yeah.
At the moment, we don't see the slides, just in case you're looking through the slides. We don't see the slide.
Yeah. Thank you, Francis. I've got yeah. Come on. Share. I apologize. Mostly what I was doing is talking. So.
Oh, there you go. That's beautiful.
Yeah. Okay. And
Great.
so, yeah. So the point that I was getting to in a very wordy way is that there's a strong decision analytics, decision support foundation for using quantitative systems pharmacology, and you might want to answer, any one of these questions. To give you an idea of a use of POP-PK in clinical, we did a model of a disease. We did some simulations, and we showed to the regulatory agency's satisfaction that a phase 2 trial was not necessary, and that a phase 3 could go from 3,000 subjects down to 700, which is a huge impact. So that's kind of like one application I'll throw out there. But all of these decisions need support. And if you don't have the specific clinical data that's relevant to that explicit decision, you're predicting something, right? How would you handle these decisions without a model? Well, you'd hire very expensive key opinion leaders, perhaps, and you'd say, "What do you think?" And he or she would say, "Well, I think you should put this drug into clinical." And the other one might say, I don't. What you're really saying is you're hiring 2 or 3 people to integrate petabytes of data about the disease, the physiology, and all of the numerical and analytical methods that have been used, and they're integrating that in their head. And I would argue that that's not perhaps the best way to do things. Maybe it's better to integrate all that information in an organized fashion, tied together with mass balances, physical laws, the QSP model, and have something that's quantitative that can support those experts so that they can meld their clinical judgment in with a quantitative prediction. So I'm showing you there the classic go no go tree for phase 1, phase 2, phase 3. On the upper point there's decisions at each point. And then right underneath that, I've showed you kind of a schematic of what we do. We take some alternatives inputs, and those inputs might be trial protocols. They might be different patient parameters, so different virtual patients or different trial arms. And then we predict what will happen for that patient in that arm, and we might iterate. And then when we get done with the trial, we compare that simulated trial result or predicted result with a desired trial result. And then we might go back and we might change the inputs to see what we get. I'm a control theorist, and my PhD work basically involved something called nonlinear model predictive control. And I would point out that the thing I just described, inputting inputs, seeing what you get, optimizing the inputs is an optimization. It's an inversion of the model. So what you're dealing with is a model inverse. So that's what we really want. We don't want to say, given a therapy, this is what would happen. What we want to say is, we want a healthy person. What therapy do we want? And that's kind of some of the goals. Given what we want, what is the best trial protocol? Given what we want, what is the best dose form? That sort of thing. And that's what we're doing here. If we're trying to decide what kind of modeling to use, sometimes it's pretty clear. I mentioned that I'm not. I've done a lot of PK models. I've done some pop PK models, but I'm not anywhere near your level of expertise. So if I've got something wrong here, chime in and let me know. But in general, we know that we might want to do NCA, non-compartmental analysis, initially to get an exposure dose. We know that we'll do possibly compartmental pharmacokinetics, usually population pharmacokinetics, and this gives us things like exposure clearance, half-life, efficacy, and covariates. And it's also a regulatory requirement typically. Back when non-mem was first involved, I think that was around 1987, very shortly after, the people that invented non-mem, very savvy, and they worked with the FDA, and at some point the FDA basically started telling sponsors, either you do the pop PK modeling or we will, and obviously better to know what's coming and do it yourself. Physiologically based pharmacokinetics, also, it actually evolved before pop PK. So I think the first papers were in 1972. That was Otto Bischoff and Robert Diedrich. Bischoff ended up being a professor at University of Delaware, where I've lectured occasionally. I'll brag a little bit. And this has been incorporated, and we'll talk about pluses and minuses of that. Quantitative systems pharmacology, you could go take the history of that back to, say, 1952. Hodgkins and Huxley, two scientists in England, figured out how to represent the action potential generation in a giant squid nerve, an axon from a squid, and they won a Nobel Prize for their insight. And to give you an idea how far things have come, they had a 3-compartment model that they did that was physiological, and they won a Nobel Prize for illustrating how action potentials work. So sometimes it's obvious what you need, but sometimes it's not. And how would we start to decide? And I would argue that really you kind of have to look at what decision you're making, how the analysis you're proposing will change that, the cost of, what is the cost differential between, or the benefit differential between a good decision and a poor decision. And I put it in orange here just to make sure it's emphasized. So often, I'm coming at this from somebody that has not only done modeling, but I've had to sell modeling for the past 25 years to skeptical and not modeling sophisticated people. And I can show them a model and they say, well, that doesn't have the same statistical strength that my non-med model is, so it's not any good. That's not the question. The question is, what are you going to do without the model? How are you going to make the decision? And how would the model affect the decision you're going to make? And that's the thing that you kind of have to stress. Um, we would also basically say, how much better will the decision be? How much more confident will we be in the decision? But then you have to consider the cost and the time required for the model, the data that you're going to, do the data exist? If my drug is based upon a theory that it's affecting pathway A, and I have no data on pathway A, perhaps rather than modeling, we might do an in vitro experiment or some other analysis to find out how the pathway A works and what affects it. Let's see. Often we are limited, and this is actually good news for those of you that are interested in doing QSP modeling, perhaps. We're often limited by the resources we have. There's not enough QSP modelers. There's not enough good QSP modelers, so you have to consider that. And here's something. The last point is something that I want to stress because it's kind of it's not often thought of. In my experience in working with American drug companies, American and European drug companies, and Japanese drug companies, they have a specific decision they have to make. You go in, you do a model, and these models are not cheap. Somewhere between $200,000 and $1 million in the U.S. to do an adequate QSP model. More typically around $400,000 or $500,000. You go ahead and you do this. You add to the decision, and the model gets tossed aside. It's a valuable resource, and there's some value in even the non-modeling portions of doing a model. So, if you're in a company and you want to take over a disease area, if you really want to continually improve, and this could happen in academia too. Melissa Hallow at University of Georgia has a very good model on circulation she's developed based upon a 1972 model of Arthur Guyton, but she's raised the level way up. You can build this model up and keep improving it with new data and keep improving it with new observations, and then you start being able to interrogate the model and leverage it for looking for new targets and so forth. Actually, I'll just share with you a big lost opportunity. I think many of you are aware of the new, the GLP, GIP treatments in diabetes that is also causes weight loss and has been a massive success for Novo Nordisk and others. We were looking at GIP and GLP in a diabetes model in the year 2000. Had we done the platform and really exercised it and really looked, I think we could have had those drugs 20 years ago. So big opportunity lost. So hopefully that's an endorsement for platform models. My company right now is working with both Merck and Genmab doing platform models in fields of oncology. So I'm pretty excited about that. Now, I mentioned that I'm an engineer, and engineers typically get very poor statistical training, and that adds to a prejudice, right? That adds to a mindset, a bias, if you will. And so I'm typically going to think of models as causal. And because of the lack of statistical training, I don't tend to default into a statistical model. And there are other people that know statistics up and down and might not trust a mechanistic model. Just a quick insight. One of my friends, an African fellow, Babatunde Ogunaike, Nigerian fellow, he and I were friends since he was a graduate student and I was an undergraduate. He basically realized engineers didn't have good statistical training. The field he was in was polymer chemistry, polymer kinetics. And if you write the equations for a specific reactor that makes polymers, all deterministic. I'm specifying the volume, I'm specifying the flow into the molecular weight distribution is a random variable. And So he realized he needed to understand statistics better to be able to characterize that. And so he ended up getting a master's in stats along with his PhD in chemical engineering. And his thinking has been very influential in my life. So the stats center view, and forgive me if I mangle this, but Carl Pearson formed the first academic statistical group in England in 1910, something like that. And philosophically, and actually even there's some religious belief that he was very, very strongly biased against talking about causality. And you've heard you can't show causality from observational data. You can only show correlation, that sort of thing. But to be actionable, you kind of want to know what the causal relationship is. If I increase the dose, what's going to happen? You want to know, you want to know what's going to cause a change in the therapeutic outcome. In Pearson's case, he was looking at statistics, and he was looking at ways to improve or reduce bias in outcome. And he had a farm field, and the farm field had various degrees of water and drainage and the soil type and that sort of thing. And he was trying to figure out, how do I chop up my field in a way that I can get an accurate view of what is important um, uh, in growing crops. It actually was, how do I spread manure? Well, that was the basic question. And he found that after doing all the experimental design, Latin squares and some very sophisticated things that he developed, found that actually a randomized approach in many cases was better. And certainly that's the gold standard we use now. There remains a problem with that. You can't really extrapolate beyond the sampled population. You can, but you're doing something other than statistics at that point. So that's an issue. And the other issue is to do an adequately powered trial is extremely expensive. Trial subjects are, in this country at least, $30,000 to $50,000 each, and it's really, really expensive. So to get an adequately powered trial to drive the statistics may be an issue. But the assumption really is that any data from outside the trial cause bias. And so we basically are forced to ignore all of biology and physiology. Jump in if I'm wrong there. You can correct me, but essentially what we would say is when we get adequate data, we can get a rigorous model that relates to relevant patients that we've sampled, and that gives us some real good power. And you have to do that, like, for example, for an approval trial. I'm not saying anything negative about statistics. I'm just saying that if you haven't done the trial yet, if you have a counterfactual situation, I want to know about something I haven't done experiments on or haven't done a trial on, you're kind of painted into a corner there. So that's one bias. And there's strengths to that bias and there's weaknesses. The other bias is a mechanism-centric viewpoint, and that viewpoint is basically, hey, physiology is a system. The system is related. There's all sorts of components in that system. We can look at the system and get data, but I have a little thing by the Belgian artist René Magritte up there. Basically, everything we see hides another thing. I thought that was kind of a interesting and appropriate quote for trying to look at a system in a whole and figure out what's going on in the system. I'm a control theory guy. If you have a system that has multiple feedback loops, nonlinearities, multiple pathways, and physiology has all those, and plus you have different states, disease states, and then you're talking about genotypic and phenotypic variation, looking at a clinical trial is and trying to understand exactly what's going on is going to be well nigh impossible. You can get input-output relationships fairly well characterized if you have adequate power, but going back to the statistics bias, but this is a really tough ask. So we also have things that we can't test. So you're not going to get longitudinal data using multiple needle stick biopsies of some poor patient's liver. You're not going to poke them in the liver with a 15 gauge needle every hour for, 48 hours. So you really can't get the data that you need in a trial sometimes. What you can get is you can break up the system into individual things. You take the cells out of the body, breaks the feedback loop. You can look what happens if you put a drop of the drug on in the petri dish. You can express receptors in Chinese hamster ovary cells and see what the binding characteristics are. There's a bunch of things you can do to reduce the system to individual little components and test it. And once you've done that, now you've got a very good, for each little component, you might have 5 or 10 journal articles, and you have the PDFs and the data available for that one little thing. So I just want to make the point, when you have a QSP model, you have a lot of parameters, hundreds of parameters in some cases, and a lot of statistically trained people throw up their hands and say, how, you can't fit that. But the point is, we're not fitting it with just a single trial data. We're fitting it with all physiological data that are available. So the actual model component models are over specified, and we can get a very accurate view from the literature. Now, are those parameters valid in vitro? Maybe not. We had a bonk. Do we have a question? Oh, I guess I heard some. Yeah. All right. So now we can integrate this model and test it. And I mentioned Hodgkin's and Huxley, the giant squid axon. They were able to get a 3-compartment model that described action potential in nerves, which is a really big thing. Another early model that you might want to look at if you're interested in looking at the history is that of Guyton and Hall in 1972. They have a circulation model, and that's the model, as I said, that Melissa Hollow at the University of Georgia has improved. So the models that we have, as I mentioned, we can integrate petabytes of data literally into these models, and we get a very good mechanistic or causal model. Now, I've tried to explain this for the past 25 years, and it wasn't until this year when I read a book called The Book of Why by Judea Pearl that I realized how I should have been selling it, how I should have been describing this. But basically, to my satisfaction, I know there's reasons that people argue with Judea Pearl about specific things. You can look at Professor Rubin at Harvard and look at their interaction. But the fact of the matter remains, to predict a counterfactual trial or experiment or anything, you must have a causal model. You can't curve fit your way to a prediction. You can curve fit your way to an interpolation. You can take a very strong statistical model and use appropriate techniques, allometry, for example, to expand it to a different person. But to predict something that hasn't been done, different population, different drug, different dose, you really need to have a causal model. So a causal model basically includes the disease-relevant components, and it can predict counterfactual trials by leveraging those components. This is the model that requires, because you can use a lot of existing data, it doesn't require a lot of external data, but to validate it in the clinical sense, once you've integrated the model, does require some effort. It may be harder to validate. And the other thing is, everybody asks me about regulatory aspects, and the reason that I think QSP has not been used in a regulatory sense a lot is that, at least in this country, when a sponsor company submits their drug package to the FDA and says, hey, here's all the data we've collected, can you approve this for us? And by the way, here's a check for here's some money. That's the way it works. You have to pay to get your drug approved, and that payment that the drug company makes kind of limits what they put in, so it's kind of limits the load on the FDA, but it also pays for the FDA's analysis. That triggers a time or a timeline. The FDA has to finish in the timeline specified. And if you give them a QSP model and say, hey, evaluate this as well, it's not viewed as a value add. It's viewed as an additional workload that may or may not really add to their decision capability. So there is possible regulatory antipathy. I'd like to see us figure out a way around that. So let's switch gears a little. That was a lot of me talking. I think you'd probably be interested in seeing a few examples of what commercial practice looks like. And I'm using this slide. My company is called Nova In Silico. It's a French company. There's about 30 biomodelers, 35 biomodelers, and about 15 software developers. What we've done over the past 15 years is develop not only a library of models, as you see on the right, but a methodology, as you see on the left, and a software platform that executes the models very nicely. So I've increased the size of this. This is normally not this big, but I wanted to emphasize one thing here. We start with if we're going to start a project with a company, we have a problem formulation. What question do you want to answer? What data do you have? What timelines are there? And we create a plan, and then we do a systematic knowledge review. So that's a literature search, and we integrate that literature search into our platform. There's a knowledge model component. That's the aspect of QSP modeling that I think I like most, in that when you do the literature search, you learn about the disease. If you bring a new person onto a project, you can give them the knowledge model you've created for your QSP model, and they can read it and they know the biology, essentially. It's a very, very powerful document and very useful above and beyond modeling. But that is the document we use to create a computational model. Then we use various means to create virtual populations that are representative of populations of interest. We validate the model using data we did not use to build the model, and that's where you do need clinical data for the integrated model, and that can be an issue. And then you do some analysis and some simulations, and you do simulations that support the decision. So that's how we work. And as I say, we've developed a lot of models. See the library on the left, and then the platform will do the simulations, and you can see the results from one of our simulations there. The platform has some advantages. It contains the model, excuse me, and it also contains the ability to create and use virtual populations a lot. So we can, once I have a model in the platform, if I want to create 5,000 virtual patients and I want to do a simulated trial with 5 arms with each of those patients, I can set that up in about 10 or 15 minutes, which is really, really efficient. So, yeah. So I've worked with other companies. I've led other companies. I'm really happy to be working with a group that is doing this stuff, and I think they're doing some very cool things right now. I think while I'm thinking of it, one of the things we have is an application program interface. We also have the ability to add applications. So we have an AI app, for example, that will take a modeling PDF and extract the model from that modeling PDF and put it in our system. So there's some very cool stuff that we can do. That's just a hint. So PBPK or QSP. Well, I view them as different, and I view them as often complementary. And I'm going to let you know that in my, well, let me put it this way. What we typically do is we create a disease model that has the physiology and a PBPK drug model. We typically don't just do a compartmental drug model. We do a PBPK model. And the revelation I'll share here is I've argued that we should just be doing simple PK models often because they're available in the literature. One of my colleagues, Claudio Monteiro, says no, we shouldn't, and my other biomodeling colleagues say we shouldn't. And here's a little humility hint. If you ever achieve a high position, I have a pretty good title. I'm a senior vice president, but there's no way I would countermand my biomodeler and modeling executive manager's view. In other words, this is a case where I think we might do it a different way, but I'm absolutely supporting what my team wants to do because they're the ones on the ground and they have to do it. And you have smart colleagues, you're kind of an idiot if you hire smart people and tell them what to do. So we have the disease model. We have a PBPK drug model, and you can see all of the different things we have typically in a PBPK model. And yes, there's more parameters in there. It's more highly parameter, and you do need data, distributional data. So there's some issues that are required there. And imagine too that you might have, for example, if you're doing an ADC, antibody drug conjugate, where the antibody has a bunch of uh, little cytotoxic payloads hooked to it. You have to have the breakdown, the release of those the drugs from the antibody. You have to model that in every compartment. So this can create a massive model. But our team has found that using a PBPK gives them the flexibility to do what they want more easily, even though it's harder to set up initially. So I think that's kind of something interesting um, that we do for every model. It's very impressive. I'm impressed with my team. But once we get to this point, there's kind of an issue. And given that I work for a French company, well, this guy's not French. René Magritte was Belgian, but he has um, this this painting is fairly famous. This is not a pipe. The point is, what we see is not reality sometimes, and what a model is not reality. So here's a question, and this is I'm absolutely honest in my uh, not knowing how to answer this question. How close to reality must a model be to be reliable? If we have a model that can replicate data that was not used to create it, great. But what if, for example, we have a model where if we're looking at the survival of patients, in the data, a few patients pass in the first few months of the trial, and our model doesn't show that. But then the model and the data agree exactly. Is the model good? I don't know. We tend to trust it, and so far I'll show you some nice agreements that we've had. But yeah, this is something. There's a philosophical aspect to the modeling that we really need to consider. I don't think I'd show this to a client. Having said that, I'm showing this to modeling experts for your interest. Um, so um, I I think that clearly, if you have something like a solid tumor in a body where there's receptors that you're targeting in lots of different tissue types, you really have to consider a PBPK model, right? Um, and there's some other concerns here. I'm going to zip through this, um, but the key thing is that if you're really interested in the distribution of drug or the holdup of drug, certain receptors will hold on to drug and then release it. You have to consider those things. Um, then a PBPK is more physiological, more accurately representing the reality. It's closer to being a pipe, if you will. Um, and and again, because we may use simple PBPK, we might have it, tumor tissue and other tissue, but we still use PBPK models at Nova. This has some advantages too, in that all of our PK models are standardized in the same type because many people might use them. The QSP model characteristics obviously are that you're representing a causal model mechanism. You can do predictions. You can predict counterfactuals. Um, you can do things like, for example, if I have trial data and I fit my model, now I can take my PBPK model and let's say for the trial I chose as an inclusion criteria something that was very narrow. I wanted to get a very, very accurate, strong p-value. I wanted to get an approval with a very strong p-value. I didn't want a lot of variability, so I didn't choose a lot of different people. I chose a very stratified, narrow, segregated focus in my inclusion criteria. But now I want to expand the use to other populations, or I want to extrapolate. So the QSP model can be used to extrapolate with some degree of confidence to other populations, other doses, combination therapies, and that sort of thing. So I think the key thing for QSP is predicting things you haven't done, and the other thing is predicting interactions, right? Having said that, it's useful to gain insight, Hodgkins and Huxley, Guyton and Hall, that sort of thing. But I think, yeah, to excuse me, to predict stuff that hasn't been simulated yet or hasn't been executed yet, trials that haven't been executed, and to look at interactions between a new drug, for example, and an existing one or a combination of the two. I think then it's been 40 minutes and I've been blathering on. Let me show you, I think. I'm gonna go to, I think, a specific. I'll go down here to a specific thing here. I think what you can see here right now is our disease library. Actually, I'll go back and share this. That's not what I want to do. I want a slideshow. There we go. This basically shows a lot of different models that we've executed commercially, and it shows the preclinical and clinical phases that we've used them in. So you can see it's a wide range of applications, and these involve simulating trials to, for example, find responders versus non-responders, or to compare a all-comers trial versus a very narrowly focused trial, or to evaluate a trial that has specific inclusion criteria or exclusion criteria, if you will. And there's a wide variety of things here. I think that a lot of things we're working on right now are oncology because it's complex, and a lot of things in oncology we're working on are complex antibody-dependent antibody-drug conjugates, bispecifics, and so forth. There's another application for QSP where the system is so complex that you're not, for example, you're not going to figure out what the drug antibody ratio of antibody or antibody drug conjugates is in all the different compartments in the body over time. It's really complex, so you have to do this. Let me zip forward to a model that I'm going to zip through some things here. Actually, hepatitis, I know there's some interest in HIV, so we did some hepatitis B infection in patients that had HIV. The modeling there was kind of interesting, and I think that there might be some interesting talks we could have. I mentioned metabolic models. My initial modeling was in diabetes. We have some models of different fat loads and lipid loads in these diseases, but it's again a very complex system that interacts, and the initial models I was working with are no way as complex as what we have now, but it's very interesting to be able to see what will happen with like statins and PCSK9 and other things. It's very cool. So. Oh, and this hyperparathyroidism. Our model is based upon a published model by Matt Riggs and Mark Peterson. Peterson at Vertex now and Matt Riggs at Metrum. And this was very interesting because it involved supplementing existing thyroid hormone with synthetic hormone, NATPAR. And the interesting thing to me that the model showed was that if you gave a NATPAR in a level dose, bone density decreased. If you gave NATPAR in a pulsatile fashion, bone density increased. So here's where we got a very, very good insight from the model. And this was actually used with the FDA to help get approval of some specific drugs. And we've taken that model and improved it. So we actually used it to look at like long-term, like chronic kidney disease effects and that sort of thing over simulations running 20 years. And I think that was kind of interesting to generate that model. That was my colleague Loïc Altave, and very cool stuff. So we've imported a lot of models, and then I think oncology. And the NSCLC one is the model I wanted to show you. Again, in many cancers, some cancers, for example, Ewing sarcoma, the cancers typically, there's not a lot of genetic variation. It's just there's an EWS FLY1 protein or gene that's spliced, and it's incorrect gene, and it produces a protein that messes everything up. In other diseases, you have a phylogeny, a clonal phylogeny that develops over time, and the tumor has some cells and they mutate, and then they have some other cells that further mutate. So you have a very different spread of clonal variability. And what you're seeing in the model results at the lower left there is we show you that there's the the creation of a clonal variation. That's the little red dots down here, and then that'll grow. But then there's a treatment that starts decreasing those things. And what can happen, for example, is if you have two clones and you kill one, that might release the other clone to grow. The other clone now may get better nutrition. So you can have clonal competitive release and that sort of thing. And these things have different susceptibilities to the drug. Obviously, I mentioned earlier, you can't do a needle stick biopsy every 15 minutes in a liver tumor. But so you typically don't have an exact representation of the clonal variation, but you can have an empirical representation of different clones. And with this model, we were able to take a lot of different data and predict different outcomes, such as time to progression, or objective response rate. Typically, progression-free survival or overall survival are a little bit more difficult in this prediction. We have done that, but because the mechanisms are all in the model, we can test all of these different drugs, which is kind of cool. And again, this is NSCLC, which has an EGFR mutant, and so it's an adenocarcinoma, and this is in stage 3B or 4. And this is what the model kind of looks like. And there's a growth factor, cell cycle, tumor growth, that sort of thing. And these are duplicated for each of the subclones. And then that is aggregated into sum of lesion diameters, typically, to give you an outcome. And then the time to progression and other outputs can be evaluated. One of the difficult things in cancer is, and it's weird, in the model, you have perfect analysis at any point in time. In other words, you can simulate having a needle stick biopsy in your patient every 10 minutes because you're not really taking tissue out. You can get an accurate readout of what the model says should be in the tumor. So if you're doing time to progression, you're taking data from clinical, and you don't know when that progression actually started. If you go like a month between visits and the tumor was shrinking, and then a month later it's growing a little bit, you don't know when that growth started. So it's kind of hard to predict, and we've had to think a lot about how outcomes are reported. Let's see if I can do this. And so we had an in vitro experiment, and that's another thing we can leverage in vitro experiments. And we've had some very good results where we've been leveraging mouse models, and they've actually translated very nicely to humans. This is in heme cancers, but this just gives you an idea of the different types of things we would look at in the literature to represent within the model. And we get a very good, accurate readout there. And once we've integrated that and tested the modeling with clinical data, it gets you even better results. So now we calibrate that specific model where all of the components represent reality, and now we have a nice representation in the Kaplan-Meier curve on the right of the different patients, and we have a very good idea of the variability of the patients. And then we basically fit the model, or we predicted what would happen to a specific trial, and this is we just basically calibrated on public data. So this shows you the model results when we've calibrated on public data, which is pretty good, I think. It's a nice representation. But then we were able to go and predict, and this was a very cool thing. We were able to predict quantitatively before we had clinical data from them the outcomes of two non-small cell lung cancer trials, one from AstraZeneca and one from Janssen. And we were able to announce the predictions before the trials were released. So you can see this is the FLORA-2 trial. This is the AstraZeneca trial, and this is how well we represented their data. And this is a Kaplan-Meier curve. So this is progression-free survival over time, represented the fraction of patients that do not show progression. All right. I think at this point I've talked for 51 minutes. I think maybe what I could do is open it up for questions right now. I will, I'm going to go all the way to the end. There we go. I want to acknowledge my colleague, Christina Lyon. Christina Lyon is prepared many of these slides in another context, and I really appreciate her doing so. And I appreciate the efforts of all of my colleagues that created a lot of these models that I get to describe to you. So with that, I'll say, what questions do you have?
Thank you. Thank you very much, Jim. I think this was quite eye-opening. Like you did mention at the start, it's not the typical field that most of us actually do working on a daily basis. So this is quite eye-opening, and I think it sets us up for quite a lot of questions and conversations. Members, I did post on the chat. If you have any questions, please, we have the Q&A section that can really help in more discussions, but also you can post within the chat window. So if you have any questions you want Jim to address, go ahead and post in the chat. From my side, I think I'll start off with a few common questions you do see coming up whenever you try to compare the different modeling approaches. One is a question of the inter-individual variability within patients. When you talk about this in population models, there seems to be this notion that you better capture IIV. For the case of QSP, and if you're really simulating a trial, how do you ensure that the models do capture this? How competent are QSP models in capturing variability across individuals? The second question comes in line with the complexity of QSP models. With the more equations you have, the more computational demand. For example, the platforms you work with, how computationally intensive are they? And
yes,
does this pose as some kind of a restriction to the application of these models? What are some of the, the corners one can take to make this more realistic if you're using a typical low-speed laptop that a lot of us actually do use? Maybe let me stop here for now, and we'll have more questions as we move on. Thanks.
Okay. Well, to reassure you, I used it was a pretty powerful laptop, but by today's standards, it wasn't very powerful. So I was able to do consulting for about 10 years, more than that, with a laptop. But having said that, answer your second question first. The platform we use automatically parallelizes our trials. So basically, we go in and the model has proposed distributions of parameters for different of different parameters, and there's a sampling to create virtual patients. But to create that, we often only have aggregate data, right? We often, like average data. And I'm going to just show you this. I think it might be useful. You can see this. This is for a bispecific antibody model. And we see that the model has 670 parameters and 92 differential equation states in 14 compartments. There's a certain number, most of those parameters are technical, like, what's the molecular weight of glucose or what's the molecular weight of the antibody, that sort of thing. Many of those don't affect the outcome. So that leaves, in this case, about 160 of them that we use literature values for 130 of those, and we calibrate 33 to mimic the variability of the average data that we have. So that's a limitation, but we actually get pretty good results, even if we're using the data. I'll go back to that non-small cell lung cancer stuff. We did not have individual patient data for that. We were using aggregate data. Having said that, we do prefer to have individual patients, say from a previous trial, or the thing about QSP models is you're not just fitting the drug, you're fitting the physiology. So there might be 5 parameters that relate to the drug and 50 that relate to physiology. If you have a trial of the same disease from another drug, you can calibrate the 50, right? So you're able to get an idea of the distributions of the parameters and the covariation of those parameters from a lot of different sources, and it doesn't have to be the specific drug. And so, I'd probably rather have my team give you a discourse on calibration of QSP models than answer inaccurately right now. But I think I can say that we get it kind of a good structure. We're confident in the structure. We can fit aggregate data, and you can tweak some of the parameters to see how that affects the variability in the outcomes. And then we really like to have individual patient data. But even without that individual patient data, we were able to predict certain things. To go back to your numerical efficiency thing, we do use, kind of an optimized integration engine based on sundials. It's optimized at the machine code level. The models are automatically parallelized. So basically, we send it out to AWS, and we're using a cloud service, and then we can send, if we have 300 nodes, we can send 300 patients out at a time. Typically, these models run in somewhere between a few seconds and 10, 20 minutes. For a 20-year simulation of a patient, it might take a bit of time, but still, if you do things right, and you have to, there's some technical issues, like when you program, you have to stop stiff integration solvers if you're going to have an edge condition. And what I mean by that is, if you're going to feed somebody or give them a dose of like a infusion over time, or even a dose, a pulse, injection, a modern efficient ordinary differential equation solvers do forward extrapolation, and sometimes what they can do is they can extrapolate right over the top, right past those things. So you'll be, if you have a model with meals, for example, and it'll be 3 meals a day, 3 meals a day, 3 meals a day. They missed breakfast. What the hell happened? And so you get 3 bumps in the plasma glucose every day, and then there's only 2. And that is when you use, for example, you say, if time is greater than T breakfast, feed. If time is greater than T breakfast end, stop feed. Actually, that would create an event. You would say, if time is greater than T breakfast and is less than T breakfast and feed. If you do that, you get inaccuracies in the start and stop time of the feeding. You have to really stop the integration. So you have to use events. So highly technical, sorry, but you have to make sure you do the modeling equations correctly. And let's go a practical matter, what I've used in the past has been MATLAB and Symbiology to do my simulations, and it's certainly adequate. In terms of generating thousands of virtual patients and doing huge clinical trials, it's probably not adequate for me. Does that answer your question? Is that responsive?
It does, and thanks a lot. My internet only momentarily broke off, but I see there's a comment on the chat here, Jim. I'm not sure if you can see the chat. So there are two questions. One is, what software are commonly used for QSP modeling? And two, you did mention clinical data use for model optimization. What is your take on using PBPK model generated data in QSP modeling? This is from
yeah. I'm trying to let's see. There's the chat. There we go. Okay, I got the chat now. Thanks. Yeah. So so the software that's typically used, I mean, we have one client, very sophisticated consulting company, that was using Copazi. It's cool. It's free. It does some cool stuff. But they were finding that they couldn't do the clinical trial simulation, and their client came to us and said, "Can we do this?" So we arranged to work with them in a collaborative way, so that make sure that they win and we win too. But we translated their model into our Jinko platform, is what it's called, and we use that. As I say, a lot of people use just pure MATLAB. A lot of people use Symbiology. That's what I used for many years. I'm a graphical modeler. I like bubbles and arrows and stuff like that. So that's my prejudice. Let's see, and let's see. I'm looking at the chat here.
So
What
yeah,
software?
I was also
Yeah.
interested in just the way you were describing what you do with meals and not meals, because often when we're working with breastfeeding babies, obviously a baby, how often does it feed? I mean, it doesn't always feed exactly when you expect, and we often try different scenarios. And some of what you were saying really resonates with how we try and anticipate what's happening there. So it's something I need to
Yeah.
give more thought to. So thank you for that insight.
Yeah, and it's interesting too. So one of my friends, I worked with Scott Seiler at Intellos, and Scott works for Simulations Plus, and they have a simulations group that does a lot of work in liver disease. And Scott is I would probably say he is the most sophisticated liver modeler in the world, and extremely knowledgeable in his field. I have immense respect for him. He is adamant that one has to have all of the very detailed physiological things going on, like in the mitochondria. You have to have that accurate to represent what's going on. You can't kind of approximate that over time. You have to have it accurate. And so that creates the issue of very stiff models, and then you need a stiff model solver. And once you've got a stiff model solver, now you're doing forward extrapolation. I remember writing in fact, we had a paper at the Page in Marseille in 2008. One of the first times anybody had ever done clinical trial simulation with, we did 600 virtual patients, sampling with replacement of the virtual patients, and then we did sampling of PK parameter distributions to get the PK. And we did the simulation, and I had to take the doses and the meals and all the times, and I put them in a vector. I sorted them, I eliminated duplicates, and then basically my model simulated from one event to the other. Tell me this, Katrina, for breastfeeding, does your model include a hunger? In other words, is the feeding driven by a random function that is driven by a hunger variable in the baby, or is it kind of just a random
I don't
time?
think I don't think we've ever considered hunger in the baby. It's much more, should it be like a sort of multiple small boluses? Should it be more like a QDS regimen? Should it be more like a continuous infusion? Can we assume 150 mils per kilo per day or not? Can we somehow, much more basic than thinking about the baby as actually a human being that feels hunger.
Yeah. Yeah. I'll tell you what, if you want to talk about this, be happy to. I do want to address this PBPK model. As I say, um, the PK issues are this. If you have what I call an autonomous model, where basically you create the concentrations and the concentrations affect a binding, and essentially there's no transporter mediated or receptor mediated drug disposition. If you have that, then I think you can generate nice PBPK or PK parameter profiles and use those very, very nicely. Um, is that responsive to the question? I'm not sure I'm answering it though.
No, that's great. No, I think I was just reflecting on how I was just making some notes there. I was just reflecting on how we've had so many wonderful conversations here, and that we almost need to arrange a follow-up to have it more of an extended Q&A on some of these pointers, because I'm aware
Yeah.
that time has marched on quite
It has. It has.
fast. So Jim, I
Let
want
me
to
offer
thank
to.
you.
Yeah, sure.
I've heard from my colleagues there was a plane that got stranded above South Africa due to a storm that's just landed that contained about
Oh,
four or five of our colleagues, and I know that they'll be listening to the recording, and they may also have some Q&A. So what I would do is we're all going to a modelling meeting in Johannesburg, and we'll speak to them and perhaps maybe follow up and see perhaps if you would be willing to donate some more of your time so that we can actually go into a bit more depth on some of the things.
I'd love that. And the fact of the matter is, I talked kind of without any feedback for an hour, and I can do that.
Oh, it
But
was
it's more
amazing.
fun.
No, it was amazing.
It's more fun to actually have the interactions and see the people's faces. And I'd love to have more conversations. So you let me know what you need, and I'll do my best to support you. How's that sound?
That would be absolutely amazing.
That sounds
Really,
lovely.
yes, thanks,
So I
Francis.
think we've probably come to the end of our allocated time. But you'll follow through and let me know when you want to chat, right?
This is
Exactly,
wonderful. And
Jim. Exactly. Jim, let's do the follow-up on when we can schedule another more extensive Q&A. We can do this via email. We
Sure.
really appreciate the time and the knowledge you've shared. And
Uh-huh.
let's follow up on email and try to confirm another day that we can have a more extensive Q&A section. And by the way, a lot of the colleagues who are on the plane, as Katrina mentioned, are quite
Yes.
deeply grounded in PBPK and QSP. So that would be a bigger group, and it will be more interactional. We do appreciate you a
Okay.
lot. Thanks a lot for your time. And I think for now, we can bring this particular meeting to the close, but let's keep the
Okay.
engagement going on.
My great pleasure. Thank you, Francis. Thank you.
Thank you
Yeah.
very
Good.
much.
Yeah.
Thank you very much.
Thanks, Katrina.
thank,
Thanks,
thank
Shasha.
you again.
Bye bye
Bye
for
bye
now.
now.
Yep. Bye bye.
Bye.